Okay, welcome to another week of Soul Seminar Online. Today we have a speaker, Lidor Shaar-Moshe. Lidor completed her master's in Tel Aviv University under the supervision of the late Hilo Frum. She studied genetic regulation of cell hydrotropism using transcriptional profiling, mutant analysis, and Arabidopsis. In 2012, she started her PhD in the Faculty of Agriculture, Food, and Environment at the Hebrew University of Jerusalem under the supervision of Sviv Peglin. During her PhD, she studied serial tolerance mechanisms with combination of environmental stress prevalence in the Mediterranean climate. Using Brachypodium as a model plant, she studied that different combinations of salinity, drought, and heat results in distinct life histories, which are characterized by unique physiology and transcriptional shifts. Since she was interested in integrating stress physiology and developmental regulation, she joined Siobhan Bradley's lab in Davis, California, and started working with tomato. In the past 3 years of her postdoc, she has been working on two major projects. The first one focuses on the developmental regulation and function of specific cell types under the stress conditions. And the second project that Lidor's talk will focuses on the conservation of gene expression profiles at cell type and tissue resolution among tomatoes, Arabidopsis, and rice. At that, you can start sharing your screen, Lidor.
Lidor, I think you are muted.
Yes, I'm muted. I'm organizing it. Just a sec. Okay. Can
Perfect.
you see the presentation? Yep. Okay. Good. Okay. First of all, thank you for the introduction and the invitation. I'm very happy and excited to share my work with you today. And I will tell you today a few insights or lessons that I've learned from a comparative work that we did on tomato, rice, and Arabidopsis root atlases. The major question that we asked ourselves in this project is if homologous cell types are translationally conserved across these plant species. So let's talk about roots and their cell types. So again, okay. Plant roots grow in different soils and conditions. They can come in different length, diameter, and have different, and can come in different orders. They have different lifespan and specialized function. Despite these huge differences in the dynamic environment that plants grow in, roots have three main functions. First, to interact with the rhizosphere, to anchor the above ground organs, and to absorb water and transport water and solutes from the soil. Root manifest these functions by a common set of cell types and tissues that are organized in a conserved way at the root apex. All roots contain stem cell niche at the root tip, here, which produces cell types along the radial axis. These cells are organized in concentric cylinders that create piles or layers of cells along the root longitudinal axis. Following production from the initial stem cells, the epidermis, cortex, endodermis, and the vascular cells progress through three distinct developmental zones: the meristematic zone, the elongation zone, and the maturation zone. The epidermal cells interact with the rhizosphere and uptake water and solutes from the soil. The ground tissue is composed of cortical cells and endodermis, which produces an apoplastic barrier to control transport to and from the vasculature. And the vascular cells include the xylem cells that transport water and solutes, and phloem cells that transport photoassimilates and other molecules. Many of these and other cell types and developmental zones are considered homologous, meaning that they derive from a common ancestor. However, the degree to which specific developmental programs are molecularly or functionally conserved across different species is still unknown. So far, I only mentioned similarities of root cell types, but there are some differences between species. For example, if you look at the number of cortical cells, so in Arabidopsis root tip, we can find only one cortex cell file. In tomato, there are 3 cortex cell files, and in some rice variety, we can find up to 10 cortex cell files. There are some differences within the vasculature. The number of proto and metaxylem files differ between different species, as well as the pattern or arches that the xylem cells are arranged in. But how do we study the developmental programs that underlie the cell type or tissue-specific functions and characteristics? In order to do that, we need to be able to detect gene expression at the cell type resolution. In the past few years, several methods were developed to address this challenge, including protoplast sorting of GFP labeled cell population, laser capture microdissection, tagged ribosome of specific cell population, aka TRAP, which I will elaborate on in the next slide, and most recently, single cell sequencing methodologies. Most of this work was done in Arabidopsis root, but in the past year, these methods were applied to different organs and species, including maize, rice, tomato, and sorghum. In our Atlas project, our group utilized the TRAP method, which is considered a proxy for translation. Miliron and Kaiser Kayala, 2 former postdocs in our lab, identify a set of promoters that are expressed in specific domains in tomato roots. These promoters were used to drive the expression of flag tag that was fused to GFP and a ribosomal subunit. Using anti-flag magnetic beads, the labeled ribosomes were pulled down, and the associated mRNA molecules were isolated and sequenced. In total, we have 12 TRAP lines that represent 11 different cell populations with distinct expression. These lines allow translatome isolation of specific cell types. For example, here you can see the endodermis and the quiescent center using a scarecrow promoter. And we also were able to isolate the translatome of developing xylem cells using the S18 promoter. We also obtained translatome of specific tissues, for example, the vasculature, specific developmental stages. Here you can see the promoter that marks the meristematic zone. And we were able to obtain translatome from the entire root using actin or 35S. So this work is currently under review in Cell, and I really hope that in the near future you'll be able to check the expression of your favorite gene at the cell type resolution in ePlant browser. This manuscript titled Nobility, Conservation, and Repurposing of Gene Function in Root Cell Type Development include several stories. We use this translatome to identify enriched cell type and tissue genes and processes. And then we focused on xylem and exodermis development and function using motif enrichment, network inference, and biological validation. We also identified a set of core genes and modules that are expressed in specific cell populations in plants that grew in 3 different environments and developmental stages. And finally, we explored the translatome conservation of homologous cell populations using 5 cell types out of the 11 that we obtained. And these 5 are common between all 3 species. So before talking about plants, we need to talk about animals, because the degree of expression conservation among tissues that derive from a common descent, aka homologous tissues, were studied much more intensively in animal research and especially in mammals. So the predominant hypothesis is that homologous gene regulatory network established the identity of homologous cell types, tissues, and organs across species. If you think about it, it is not a very conservative hypothesis, considering that some of the animals that were included in such comparative studies are separated by millions, or up to some up to 450 million years of evolution, as in the case of human and fungal or pufferfish. Despite this huge evolutionary distance, a study that examined the expression conservation of homologous tissues of human, mouse, human, mouse, chicken, frog, and pufferfish found that tissues clustered based on cell identity rather by the species of origin. In this heat map, which is a graphical, this heat map is a graphical representation of all versus all matrix, correlation matrix of expression profile between the 20 tissues in each of the 5 species. And you can see that the cell types are clustered together here in the middle. The alternative hypothesis is that different gene networks achieve similar cellular phenotypes, and several studies have shown this phenomena. So beyond the pure interest in basic science, the degree of expression conservation has an important practical implication on the utility of model animal species for biomedical research. If the expression conservation between human and mouse or human and monkey is low, what is the point of using model species for biomedical research? Going back to plants, this question also has broad practical implication, including utilization of model species to improve crop performances, or the use of known developmental marker to determine cell identity in single-cell sequencing methodologies. So in order to test these 2 hypotheses, we use the translatome data from these 3 evolutionary distinct plant species and just to give you an idea of how they diverged and different they are. Monocots and dicots are separated by up to 180 million years of evolution, and Arabidopsis and tomato were separated something like 150 million years ago. So in collaboration with the lab of Julia Bailey-Serres from UC Riverside, we generated and collected translatome profiles of the endodermis, here in blue, which includes also the quiescent center, the entire vascular tissue, the meristematic cortex, and the entire meristematic zone. And each of these 4 cell populations has similar promoter expression domain among the 3 species. We also have used 35S in order to obtain the translatome profile of the entire group. So we started the cross-species analysis by processing and normalizing the data that we obtained either with RNA-seq for tomato and rice, or with microarray for Arabidopsis. And we then carried out 4 types of complementary analysis. In the first analysis, we examined similarities among translational profiles. We did that based on 3 different orthology maps, and then we examined the clustering patterns with principal component analysis. As a reminder, orthologs are genes that diverge after speciation event, but the gene and its main functions are conserved. In the second analysis, we identified conserved cell type specific genes. And for that, we constructed a 4th orthology map. And that is based on sequence homology coupled with expression correlation. And in the third analysis, we applied a different approach. The first 2 analyses are based on orthology, as I mentioned, and we decided to use an initially independent, an analysis that is independent of the orthology relationship. In this analysis, we compared enriched genes and functions between homologous cell population. And in the 4th analysis, we examined the function and the degree of expression conservation, function and the degree of overlaps of genes with low expression variation across cell types within each species. So in the next few slides, I'll guide you through each of those for analysis. So we started our first analysis of global translational patterns by examining each species individually. Before we compared between species, we want to make sure that the samples are clustered based on cell identity. So in these PCA plots for each species individually, each dot represents a biological sample, and it is colored by the one of the 4 cell types that we were using. These PCA plots confirm the expected clustering pattern in each of the species, except for rice vasculature. So we also looked at expression patterns of known cell type markers that are critical for root development, and found that they show the expected expression pattern. So with these 2 confirmations, we continue to compare expression patterns between the 3 species using 3 different orthology maps. In the first orthology map, we included only 1-to-1-to-1 orthologs based on sequence homology. And in this PCA plot, each dot represents the mean of each cell population that is colored based on the its cell identity. And the different species are indicated by different shape of these different shapes. And based on this orthology map, we found that the samples of the meristematic zone are separated from the other cell population. We also used an additional map that is based on sequence homology to Arabidopsis and found similar clustering pattern. The third orthology map also showed a similar clustering pattern, and this orthology map is based on sequence homology and expression correlation of published tomato, rice, and Arabidopsis transcriptome. So each of these three independently derived ontology maps suggest that the meristem translatome is more similar across the three species than the other cell population. To gain a better understanding on the similarities between homologous tissues, we focused on genes that are conserved, genes with conserved cell population specific expression among the three species. We started this analysis by constructing an additional fourth orthology map based on expressologs. And so what are expressologs? Well, expressologs are putative functional orthologs. These are genes that have similar sequence and similar expression pattern. To identify these expressologs, we first assigned genes into families based on proteome similarity. And next, we examined each gene family separately. So, for example, in this gene family, we have one tomato gene and 3 rice genes. So we calculated the correlation coefficient values between the expression of the tomato gene, the 3 rice genes across all homologous tissues. And then we chose the gene pair with the highest correlation value and defined them as expressologs. We parsed this expressologs list and included only highly confidence expressologs with positive correlation values. The expression of these expressologs subset nicely clustered by cell identity, nearly separating each cell population. We use this map and an ANOVA approach to identify genes with conserved cell population specific expression. So in this heat map, each row represents one of the 35 conserved expressologs, and each column represents one cell population. The values are scaled across each row, and each row is the mean expression value of the expressologs among the 3 species. And so yellow indicates higher expression and blue indicates lower expression. Most of the expressologs have conserved expression in the meristematic zone in line with the previous analysis. And these genes include genes that are known to be involved in cell division, RNA synthesis, and nucleocytoplasmatic transport. I'll continue to the third analysis. And here, as I mentioned, we decided to use a different approach. Orthology-based analyses are very useful, but they suffer from a critical caveat because they are limited to the number of genes that are conserved among the 3 species. So you basically lose a lot of information. You lose a lot of genes. To circumvent this limitation, we identified genes with enriched expression in each cell population within each species. So these heat maps of gene expression show genes that are enriched in the meristematic zone, as indicated by the green to yellowish color, and each row is 1 gene. Next, so this is just one example for the meristematic zone, but we have lists of enriched genes for each of these cell types. Next, we performed enrichment analysis of this gene list using both MapMan and gene ontology terms to assess the functions of this gene list. In this heat map of significant MapMan terms, each row represents 1 term, and each column is the cell population within each species. Gray color indicate non-significant terms, and the blue to yellow colors indicate significant terms, and values are -log⁻10 FDR. This heat map gives a sense of the overlapping terms among both homologous and non-homologous cell types, but to better visualize these overlaps, we used circus plots. And in this plot, each cell type or cell population is indicated by a different color, and the number of enriched terms is in each species is represented by a separate segment of the circle. The width of the ribbons is proportional to the number of overlapping terms. This plot clearly shows that the meristematic zone has the highest number of overlapping terms. We also tested the overlaps of expressologs among the homologous cell populations, and we found similar patterns to the enriched terms. These results are in line with the first 2 analyses, and they demonstrate functional equivalency of the meristematic zone between the 3 species. We decided to use both GEO and MapMan annotation because, well, GEO is widely used. Most of us use only GEO, but MapMan annotation or MapMan tool, which is a plant-specific tool, was developed originally for Arabidopsis, and using these annotations, we were able to gain more information about the function of the genes. And we found this tool very, very useful. And in the last analysis, we focused on genes that are expressed in a similar level across all tissues within each species, as indicated in this heat map based on the similar color of each row. So this heat map of expression, each row is a gene, and the tissues are at the x-axis. We term these genes constitutively expressed genes, or CEGs. A comparative study in mammals demonstrated that genes with low variance are enriched for housekeeping functions. These functions, these housekeeping functions, occur in every tissue and are important to maintain cellular function. They include RNA binding and processing, and chromatin organization. Another study, also in mammals, showed that housekeeping genes tend to evolve more slowly than tissue-specific genes. And we were interested to know if similar enrichment of housekeeping functions could be identified in constitutively expressed genes of plants. We first identified genes with fold chain lower than 1.5 across all cell population within each species, and you can see how these genes are, what's the expression of these genes look like. And then we tested the overlap. We then performed enrichment analysis and tested the overlap of this enriched term. So this circus-based plot shows the number of enriched term within each species and the proportion of the overlaps either between all three species or between two species. In concordance with the literature, overlapping ontology terms are involved in housekeeping functions, including protein metabolism and modification, RNA binding processing, and chromatin organization. Lastly, we compared the constitutively expressed genes here, the CEGs, with the cell type enriched genes and found a larger number of overlapping terms in the constitutively expressed genes, even when we considered only the meristematic zone enriched genes. These results suggest that the expression patterns of the cell types enriched genes are more affected by speciation than constitutively expressed genes. So with that, I would like to summarize my talk and provide some conclusions. So the root meristem is molecularly homologous across plant species, and that support the hypothesis that homologous gene regulatory network establish the identities of homologous cell types, tissues, and organ across species. In addition, the similarity of the root meristem is in line with animal studies that showed a higher expression correlation at early developmental stages than mature homologous tissues. Next, the low similarity among other cell populations suggests that distinct molecular programs can give rise to similar cellular morphologies. So this explanation may apply to endodermis. Endodermis is a single cellular that contains the hydrophobic casparin strip, which function as an apoplastic barrier. And despite the conserved morphology across all three species, very little expression similarity or overlaps were observed. The next functions of constitutively expressed genes are more conserved than those of cell type or tissue-enriched genes, and as found in animals, they are enriched in housekeeping functions. And finally, the higher order properties of cell regulation, such as emergent property of gene network architecture, are evolutionary conserved between plants and animals. So the term emergent property refers to the new function that arrays when individual entities interact. For example, only when phospholipids interact, they create a two-layer cell membrane that is essential for life formation. Similarly, the function of the regulatory network could not be understood by observing individual genes, but instead from the patterns that result from aggregation of regulatory links within the network. And these aggregation patterns these aggregation patterns are observed for the root meristem and the constitutively expressed genes as were recorded in animals. I would like to finish by thanking my great friends in the lab, and especially to my PI, Siobhan Brady, to Kaisa Kayala, former postdoc, and she's currently a researcher in Utrecht, and she established this project, and to our awesome collaborators. And if you are interested in our work and want to read more about the tomato root atlas and the lines that we generated, you can check out our paper. And hopefully it will be published soon. And yeah, that's it.
Awesome. Thank you so much, Lidor. That was a lot of work, and it's really cool to see all those results, and I love those animations. So we are now open for questions. So if you have questions, you can type them in the chat. If you want me to read it out, please start with the exclamation point. I will send all comments and questions to Lidor later, so she can read them later as well as comment at them now. So Pat has a couple questions. So Pat, if you want to unmute yourself.
Sure. Hi. Really beautiful work. Wonderful stuff. I had a couple questions though. One of your selected tissues was the combination of quiescent zone and endodermis. And that's rather surprising because they're not that they have overlap, but they're not that similar in terms of their structure and function. I mean, one's a set of stem cells and one is a highly differentiated cell type that is along the entire shaft of the root. So can you tell me more about why that was chosen?
Yeah, so this is basically the characteristic of the promoter that was used to, that we used. So the expression domain of scarecrow promoter includes the endodermis, but it is also expressed in the first tier of quiescent center. In Arabidopsis, there is only one tier, only one cell file, but in tomato, there are a couple, and in rice, there are even more. So in Arabidopsis, it includes all the quiescent center, and in tomato, it includes one tier, and also in rice. So this is just the expression pattern of the promoter.
So you couldn't find one that was strictly expressed in the quiescent zone to use? I mean, that would have been very interesting.
No. We have also promoter that is expressed WOX5. It's a promoter that is expressed in the quiescent center and the initial of the vasculature. So we use, in order to infer the cell type enriched genes, we use we kind of like subset the expression patterns using different promoters. And so we were able to kind of like, in a way, exclude genes that are expressed only in the quiescent center from those that are expressed in the endodermis and the quiescent center, and then focus on the endodermis, basically, mainly on the endodermis.
Oh, okay.
We cannot separate endodermis and quiescent center.
Okay, the other question I have is that I was really surprised with your results from the CTEG that there was so little overlap. How do you explain that result? I'm very surprised at that result.
Okay, so we were really puzzled about this result, and well, it depends on which tissue or cell population you're referring, because if you're talking about looking at the meristematic zone, so something around between 25 to 30% of the enriched terms overlap with other with terms from other in meristematic zone from other species. And so there are a few steps or caveats in the way. One is that the annotations for other species, except from Arabidopsis, are not great. So we were limited by this step, the annotation step. And this is why we chose to use both Geo and MapMan, kind of to have a more comprehensive understanding on the gene functions. And then, so I think this is something I didn't talk about in the work, but in this presentation. But this work focused mainly on similarities, not on differences. And so the main reason is that the data from each species is coming from either a different lab or was done in the same lab, but in different years, different platforms, different people. So there is this confounding batch effect. And although we account for that, we cannot directly, or we are more confident. We have more confidence in the similarities that we have rather than the differences. Because the differences, in a way, can be related to technical effects or the batch effects. So the meristematic zone, we are very confident about what we saw, about these similarities. But for the other tissues, we decided not to focus on the differences, but we suggest that it could be that the alternative hypothesis of different gene regulatory network establish similar phenotype. This hypothesis could apply to other cell populations.
Okay, thank you.
Awesome, thanks. Federico, yeah, do you want to ask your question?
Hi, very nice talk. I might be wrong on this, but it seems that in some of your analysis, there is more similarity in the transcriptomes of homologous types between rice and Arabidopsis than between Arabidopsis and tomato. Do you think, is it the case? And what do you think are the causes of that?
So I'm not sure the causes are biological. Yes, we noticed that, and we talked about it, and we tried to make some sense out of it. I'm not entirely sure it has to do with the biology. It could be just annotation or something that is more technical. And when annotation will be improved, we might, if I don't know, 10 years from now, I'll run this analysis again with improved annotations. I might see an even better overlap.
It's kind of rice and Arabidopsis have actually better annotation that tomato, and maybe that's causing that. Yeah. Okay, thank you. Thanks a lot. Very nice talk.
Is there any other questions or comments? You can unmute your mic. You do. So what's the next step for you, Lidor? Are you going to do some more experimental fieldwork stuff? Or are you going to do more work?
I'm really hoping that I'll have the opportunity to use the data that we gained from, or the insights that we gained from this project about cell identity and conservation of expression, and apply it or use it for single cell sequencing experiment, because I have a good friend now that is a postdoc in the lab that is started to do single cell sequencing in tomato. And this is not trivial at all to assign cell identity. So I'm hoping that using the data that we obtained here, it might improve or help to run single cell analysis.
Awesome. That's awesome. Rocio, I think, has a question.
Yes, thank you for the great talk. It was super interesting. And it's just a very probably silly talk, a silly question that I have, because I wonder why those three models, why did you choose them in the first place?
These plants or these tissues?
Sorry? Oh.
Why these three plants?
Yes, these three species as models.
It's basically just because only these three species have this type of data. Other species and additional cell types have transcriptome data, but only these three species and specific cell type have this translatome data that I could use and compare.
That's great, thank you.
Awesome. Well, thank you so much for the talk and coming so early in the morning to present your work. We really appreciate it. We love learning from everyone, especially when we get to learn something new. So next week, we're actually gonna have our own Andreas presenting. He's gonna be presenting Roots in the Clouds, Evolution of Epiphytism and Solanaceae. So hopefully we get to see you guys all next week. All right, have a good weekend.
Thank you so much.